The nucleosome is the unit by which DNA is assembled in eukaryotes. And as you can see, the first step is the formation of the histone octamer using the core histones. And that looks like beads on a string when you sort of step back and look at it as a whole. Both of these forms are active in terms of transcription. But the next step that kind of closes it down a little bit more is adding histone H1, that one that goes between nucleosomes and condenses the DNA. And these 30 nanometer fibers are less active. So we have a range of activity in terms of gene expression that can happen at this stage of assembly. And it turns out that there are proteins that modulate whether or not you condense the DNA further. And that is whether they allow this close packing of nucleosomes. And those proteins are called barrier proteins. And they confine the assembly to specific areas and impede the folding of nucleosomes into these higher order structures that are more compacted and less available and therefore less expressed. Now it turns out that barrier proteins also play a role in separating segments along chromosomes that may be involved in different activities. And what we're going to look at is histone variants that are involved in forming the centromere. This is a stable, condensed structure even when the rest of the chromosome is open. And this is the point at which microtubules and other things make contact. So we have normal nucleosomes and nucleosomes with these centromere-specific histone H3 molecules. And they have different properties. They interact with a sequence-specific DNA binding protein to form this centromere-specific structure with the H3 histone involved in the nucleosome structure. And that sequence-specific binding protein also binds to the microtubules that interact with the yeast kinetochore when you pull chromosomes apart after replication. So chromosomal structures can sometimes be inherited, so that if you get alterations in H3 that recognize different regions, then you can sometimes get a second centromere at another site, or the centromeric site can move. So there are multiple processes in which histones are involved in addition to just packaging the DNA. Now once you form this solenoid structure with H1 and you've got these really much more like closely packed beads on a string, during interphase these are packaged further for the actual division process of the cell. And you add scaffold proteins to get this folded solenoid structure that is more compacted, and then as we'll see later there are more scaffold proteins. So to assemble chromosomes, we started with the naked DNA, we've gone to nucleosomes, we've gone to the solenoid structure, and to get to that solenoid structure there are specific proteins that are involved, and they're called SMC proteins, and they sort of have this hinge structure so they're able to latch around and, if you will, knit the DNA together, and they assemble into higher order structures themselves. As you can see here, they first bind and then they form this 8-membered ring, and then those rings stack on top of one another to give the very condensed structure of chromatin just before cell division. So they form the folded solenoid, and then it condenses further, and finally you get the mitotic chromosome. So we know relatively little about these additional scaffold proteins that form the highly condensed chromatin that is characteristic of the mitotic chromosome. So why do you think assembly of DNA into chromosomes is important? And what are the key steps in this process? And then, most importantly, how does the cell manage access to its genetic information? Because that's where the game is. What proteins, what regions of DNA that encode a protein get to be expressed?